Hello, everyone, and welcome to episode 27 of Diabetics for Diabetics Radio. Today's episode is titled Iron and Mitochondria. We are tracing iron's path through a diabetic's cellular energy system. I am your host, Bowie. I have had type 1 diabetes for 22 years. This slide has not been updated. I just celebrated my 22nd diabetiversary this past August. I am healing my diabetes naturally and sharing information as I go. About 2 years ago or so, I decided that I didn't want to have diabetes anymore and that I should take a closer look at what we knew about it and the information being disseminated and if there were any possibilities for that to change. And everything I've found, I have shared here, either on TikTok or via the podcast here on YouTube. And I'm finding a lot of exciting things, a lot of positive things, and I have hope. If you haven't collected that through the material yet. But today's episode is dedicated to mitochondria because in a post I made on TikTok a few days ago, I revisited how prevalent iron was in each of the theories surrounding diabetes. I mentioned parasites, I mentioned nutrition, and I mentioned mitochondrial health because there are a few people talking about how mitochondrial dysfunction in the pancreas, in the beta cells, is a source of dysfunction and part of the reasons why beta cells die. And I knew about the mitochondria theory behind diabetes, but it had been a while since I revisited it, and so I took a closer look at the mitochondria, and it just reinvigorated my passion for the iron overload theory behind diabetes or aspect of diabetes. And so here I am presenting on it. All right, so before we get started, we have to know what our mitochondria do in order to know what dysfunction looks like, right? So everyone kind of remembers their 7th grade biology teacher kind of, pumping their arms, showing their muscles, and telling you that the mitochondria are the powerhouse of the cell, right? This is like the engine of the cell, site of cellular respiration, the electron transport chain, where glucose is brought in and converted into ATP, which is the kind of energetic currency of your cell. Interesting facts about the DNA about mitochondria is that they have their own DNA. It is a circular DNA. It is believed that mitochondria were at one time bacteria independent of prokaryotic cells, and that through it's called the endosymbiosis theory, mitochondria entered the cell, they became symbiotic, and that the mitochondria was just kind of assimilated into the internal structures of the cell itself. And so here we are. Now, mitochondria, they're called the powerhouse of the cell because they act like a power plant. They are taking in imports of glucose from our food, as well as fatty acids, right? So they're taking in basically the products of our food. They are brought into the mitochondria. The mitochondria is taking them through the electron transport chain, oxidizing fats, and converting glucose into ATP. And in that process, they are packaging and offloading waste within the cell to be taken care of. We'll learn more about what that waste looks like at their reactive oxygen species. And there's live feedback, right? We'll talk also more about how the mitochondria decides the fate of the cell because there is live feedback with the mitochondria and what's going on outside of the cell. And that is determining how the cell thrives, if the cell thrives, how many mitochondria are needed, things like that. For the visual, right? So we know in the center, kind of surrounding my head here, that's the mitochondria, right? And over here, we're taking in oxygen, which is like an accelerant. It's like an accelerant. It's a pro-oxidizer. We're taking in glucose, the glucose and the fatty acids, which are going to be oxidized within the mitochondria in order to put out energy and also offload waste. Okay, now because the mitochondria is so important in creating energy, the entire cell runs on energy. If the mitochondria is unwell, the cell is unwell. And in states of dysfunction, in kind of irregular cell death, which is what people with diabetes are experiencing because beta cells are dying off, if the cell is unwell, the mitochondria are unwell because they are unable to create energy efficiently, or cellular conditions are such a way that they are telling the mitochondria, hey, time to stop creating energy. We're not able to use it well, right? So if the mitochondria are affected, it's basically kind of like taking it off of life support and turning the lights out. Okay. Now, how do we know that this kind of coincides with diabetes? Like, how does this actually work? So, if you're familiar with kind of how I've connected iron overload to diabetes, we'll see after this diagram here kind of how influential iron is in mitochondria and how it's been tied, even without mentioning iron, to the diabetic state. So there are basically three possibilities, three exports from the mitochondria. The first is the waste, right? You see kind of these are the four different complexes within the electron transport chain. So after the first complex, everything goes through. One of the production offloads is hydrogen peroxide. Hydrogen peroxide is a reactive oxygen species. It's just a waste byproduct of the oxidation of glucose. Now, how mitochondria signals for apoptosis is if it is in a state where, things are not going well. It could be a backlog of electrons through the electron transport chain. There may be some sort of mineral or tool that is not able to be used effectively throughout the electron transport chain. If there is a backup, if the cell has some sort of chemical signal telling it that it is not safe, what will happen is it opens up these huge portals in the outer membrane of the mitochondria, and basically the mitochondria dumps the contents of its insides into the cell. And the most significant of those contents in the mitochondria that is not outside of the mitochondria is cytochrome C. And so the cell knows that if it is sensing any sort of cytochrome C, it knows the mitochondria has dumped its innards and it's out of there. That's a signal for the cell to kill itself off in order for the health of the cell itself. If that doesn't happen, right, and everything's kind of flowing smoothly, the electrons eventually get to complex 5 of the electron transport chain, and the final product is ATP, right? And it's distributing ATP throughout the cell. in order for it to do its job. Now, how does that tie in to diabetes? Well, there was a period of time where, just like I mentioned before, the mitochondrial health and its determination of cell health was being applied to the beta cells of the pancreas. So everyone was like, okay, well, beta cells are dying off, so that must mean the mitochondria of the beta cells are unwell, and that's why diabetes forms. So you have all of these papers being, and there are some even before these. These were the titles that kind of hit it most on the head. But people were like looking at and getting the genetic material of the mitochondria within the beta cells and saying, well, how does this line up? I think one of the papers that I found on this mentioned that there were some consistencies in the mitochondria of people with diabetes in terms of genetic mutations, and it ended up being somewhere between 0.5, 1%-ish, all the way up to 10%, depending on the type of the diabetes. Now, was something wrong with the mitochondria of the beta cells, right? Was that kind of the origin of the dysfunction that led to the beta cell being killed? Was it genetic? Was it environmentally? What ended up pulling the plug? And in terms of order of operations, was it faulty mitochondria that led to insulin resistance, or was it insulin resistance that messed with the mitochondria being able to utilize or function? Because one of the things, and I'll see if I can go back here. Okay, with this, right? When we eat, glucose is getting into the cell and being used by the mitochondria. If we do not have insulin, glucose, or not using insulin properly, as in insulin resistance, glucose is not getting into to be used by the cell and the mitochondria, right? So insulin resistance, if that's happening beforehand, is going to affect the output of the mitochondria. So in the research, that was one of the topics that they were trying to discover. Now, they were able to find bits and pieces supporting either, right? So there were genetic ties, but they were not consistent, either via the type of diabetes, whether it be type 1, type 2, type 1.5. There were even mitochondrial diabetes. There's so many more diabetes than I care to know, but the impact that the mitochondria had on the development of the diabetes was inconsistent, even across ethnicities. One of the papers was in a Korean population. They took what they found in that and realized that it was not consistent in European populations. So it was a little bit iffy, not enough to come right out and say, hey, it starts in the mitochondria. Another paper found that cell mitochondrial function was positively correlated with insulin receptor availability. Now, it's not worded very well on the slide, but it was saying that insulin resistance was the leading factor in cell mitochondrial function, right? It was really flipped. So the more insulin receptors were available, the better the cell functioned. Now, that goes hand in hand with this slide, right? So if you have insulin resistance here, of course the mitochondria is not going to function because it's not being fed glucose properly. So the research was not super conclusive. But when you reason and you look at the bits and the pieces of how a mitochondria operates, possibilities start to open up. So when I looked at this, I thought, if the beta cell dies, that means the mitochondria had to have signaled for that to happen. Or even if that were true, let's just entertain it. So what are the things that cause mitochondria to signal cell death? Right? We mentioned before the cell opens up and offloads all of its contents. But what are the things that slow a mitochondria down enough to get it to the point where it says, hey, it's not worth it, off the cell? One of those things was ROS toxicity. Now, mitochondria are not unfamiliar with ROS. We mentioned before, reactive oxygen species are a byproduct of typical, regular, normal mitochondrial function, right? It's not unfamiliar with ROS, it's just the degree and the quantity of ROS. Now, some papers suggested caloric surplus and inactivity. The obesity model behind type 2 diabetics suggested that because of the overabundance of glucose in people eating too much, oversupply the glucose, it inadvertently created too many toxic byproducts, too many ROS, because the mitochondria were running through all of the food that's being supplied in an obese state. Now, okay, possible. Even inactivity. So it's well documented in the exercise science realm that increased aerobic capacity, so those that exercise and train aerobically, running, HIIT training, what have you, that increases the amount of mitochondria density in the cell. And so you have more mitochondria, or rather the opposite of training and increasing the density of your mitochondria is that if you do not train enough, you don't have a lot of mitochondria. And that combined with that caloric surplus where you're eating a lot of food, right, supplying a lot of glucose and creating a lot of toxic byproducts, that combined with the fact that you don't have a lot of mitochondria to do the processing of that glucose causes a backup of glucose, a backup of mitochondria, and the electron transport chain is not able to efficiently move through everything. That backup is enough to enough to cause the mitochondria to signal for cell death. And then my own 2 cents in mineral balance, and it's not just my 2 cents. There is mention of reactive oxygen species toxicity as a result of iron overload. So like I mentioned before, overeating and undermoving is like overstuffing a furnace, right? So you're compromising furnace efficiency in being able to put out ATP, but you're also increasing the likelihood of the fire itself going off. More food, more toxic byproducts, less activity, less energy, and fewer mitochondria needed. The breakdowns in the efficiency of electron transport chain increases the production of ROS. Increased ROS signals for the mitochondria to signal cell death. But when you look at this list and you think of all of the things that are an important part of mitochondrial health, vitamin C as an antioxidant to counter the normal oxidation and ROS output, zinc, calcium, sodium, and potassium as kind of your basic cell biology, sodium potassium pumps influencing cell health, B vitamins, magnesium, iron, and sulfur clusters are all a part of typical mitochondrial health in basic cell membrane fluidity, calcium-induced neurotransmitter release, and what have you. Now, when I look at these lists and I look for things that trigger my kind of like spidey senses as to something that could be going on, what do you think I see? I see iron, and I see reactive oxygen species present in the same place. What does that make me think of? Anywhere iron and ROS go, the potential for chaos follows. Because if you will remember from episode 12 of Diabetics for Diabetics Radio, where I talk about how iron influences inflammation, we know that free iron is able to weaponize typical ROS production in the cell via the Fenton reaction and the Haber-Weiss reaction into an absolute mess. Remember, it takes that typical, that usual hydrogen peroxide byproduct and turns it into a superoxide anion, hydroxyl radical. Bad news. I even gave you the graphic for it here. So, hydrogen peroxide, usual, typical byproduct of the electron transport chain within the mitochondria, combined with iron, could be an issue. But you may be saying to yourself, okay, yes, you have abundant reactive oxygen species. Yes, iron has the potential to be influential in making that ROS more dangerous. But Bowie, how much iron is even in the mitochondria? Enter the fact that mitochondria are major hubs of iron utilization and accumulation in states of health and in states of dysfunction. Because looky here, in lung carcinoma cells, that's a cancer cell, approximately 20% of the total cellular iron was found in the mitochondria. Interesting. Why is iron accumulating in the mitochondria? In rat hepatocytes, liver cells, the concentration of metabolically available iron in mitochondria was twice that in the cytosol, which is the kind of like outside, basically in the cytoplasm. So that's to say that, okay, Bowie, how much iron is even in the mitochondria? A lot of iron is in the mitochondria. That really sets off my spidey senses. Because as we know, lots of ROS and lots of iron is cause for concern because of the chaos that is produced when free iron and all of those ROS start to overaccumulate. So in a typical healthy cell where iron is being correctly utilized and distributed, not an issue, right? Because of the iron sulfur clusters within the complexes of the electron transport chain, everything's being effectively utilized, everything's being effectively distributed and disposed of, both the ROS and the iron. But in states of dysfunction, like we mentioned in the state of obvious dysfunction, lung cancer cells, there is, in those states of dysfunction, iron is present. ROS and inflammation, as suggested in the fact that it is a cancer cell, are also there. So what more needs to be said, right? What more needs to be said? There is a huge potential for inflammation in states of imbalance when you have too much iron and too much ROS, as evidenced by the state of dysfunction where mitochondrial cells or mitochondria within the mitochondria have way too much iron and way too much ROS. So what, in the grand scheme of things, does that mean for us? Rebalancing iron metabolism always requires adding in what's missing and taking away what's overabundant. So, what are the things that can balance and avoid that overabundance of iron and the overaccumulation of ROS? Copper, magnesium, glutathione is a huge naturally occurring antioxidant, specifically within the cells and within the mitochondria. Potassium as kind of like that lagging factor to accompany the sodium, right? Typically when we think of an overabundance of sodium and too much sodium, we know that that's bad. But an overabundance of sodium is also an indication of an underabundance of potassium, right? So we mentioned vitamin C before as a counter antioxidant to the oxidation that is occurring, as well as fat-soluble vitamins, vitamin A, vitamin E, vitamin K, that all serve antioxidant properties like vitamin E, as well as iron-regulating properties like vitamin A and vitamin K. So in with all of those and out with the processed foods, because of the caloric density, because of the pro-inflammatory conditions of removing the magnesium, removing the copper, removing the trace minerals and the fat-soluble vitamins, as well as adding the reduced iron that is accumulating and inducing extra inflammation. And I might just add in here my 2 cents, don't supplement calcium, because you are getting enough calcium in the foods that you are eating, and too much calcium, too much iron, not enough of any of the others exacerbates the inflammation, exacerbates fibrosis and calcification, which all result when cells die off and apoptosis is induced. Okay, so a little bit shorter episode here. Thank you for being here. Thank you. Check the description for the supplement brands that I am using to take in copper, magnesium, glutathione, potassium, vitamin C, and all those fat-soluble vitamins, as well as nutrition guides, supplement guides, exercise routines. Also check the description for literature citations for all of the papers that I mentioned, including these as well as the other ones talking about the connection between mitochondrial health and diabetes. Okay, so this is episode 27 of Diabetics for Diabetics Radio, Iron and Mitochondria. I'm your host, Bowie. Thank you, and enjoy the rest of your day.